Hi, this video is to help you with the empirical and quantitative skills assessment worksheet that we have. You do as part of the lab portion. And so just to start with, I am going to show you some of the equipment that would be used in doing this type of an experiment. We have just started in the fall of 2024 because we just bought the spectrophotometer, which is what this equipment is. And we're starting to incorporate it into the lab portion. For the online portion, we have you do the worksheet. So this will explain how we got the numbers on the worksheet, hopefully. So this is a spectrophotometer. What this machine does is you can put samples in these little spots right here, and I have a close-up of that in just a moment. And as you can see, we've got a screen here where we'll show numbers of the readings. What we are doing is we are putting a sample in here. There's a light source in here. There's going to be a light that's going to shine through the sample. And it's going to measure the amount of light that makes it all the way through the sample and out the other side, essentially. Remember, bacteria, when they grow in a liquid media, they turn it turbid or cloudy. So the basic premise or idea behind this is that the more bacteria growing in a broth, the cloudier it's going to be, which means the less light is going to pass through. Now this machine, we can take a couple of different types of readings. You can do what's known as percent transmittance. That's how much light actually passes through. So if you're measuring percent transmittance or percent T, you would expect the more bacteria, the lower that number is going to be. Now, what a lot of people do, they feel it's more accurate to measure the percent absorbance, which basically is the inverse. How much of that light is absorbed or does not pass through. And so it's inverse of the percent transmittance. In other words, it's going to be opposite. And so typically that's what we record is the absorbance. So that would mean the number is going to go higher because more light is being absorbed and it is not allowed to pass through, the cloudier the substance is. So this is just showing right here what the spectrophotometer looks like. That's just showing a closer up view of the little dial. This is a nice piece of equipment. We can adjust the wavelength that we measure when we are measuring for monitoring growth of bacteria. We tend to measure that at 600 nanometers. We can change that wavelength so that we can measure protein, or we can measure DNA, or there's a huge wide variety of different experiments that can be done using the spectrophotometer, not only in biology but also in chemistry. And this is from the manual for the spectrophotometer, just very briefly showing you've got your light source. It's going to go through. Here is your little sample compartment. The light's going to go through, detect, and we take either that percent transmittance. 100% would be that all the light passed through. And that's going to be the same thing as zero absorbance. If all the light passed through, it's zero absorbance. Like I said, we're going to be reading the absorbance, or the A number. It prints out both numbers when you put the sample in. So our particular spectrophotometer has actually spots for 4 samples. It is this closest spot right here where the sample is going to be read, and you can just slide this holder into place. It clicks in place if you want to put 4 samples in and quickly read them at once. I'm just showing you what it looks like. And the light is going to shine right through here. There's a slot in the side that holds your sample, reads through, and then on the other side, how much light got through. This is a cuvette. It's the holder. This one holds 1 ml, so we put the sample in, and it's just going to read right through here. And this just shows, if you can tell right here, the cuvette is in place. So that has a sample in it. You wipe down the sides with lens paper so you don't have any fingerprints that are going to interfere with it. So that is the setup for the spectrophotometer. Now the idea is you're trying to figure out how many bacteria are in a sample. What's that concentration? You've seen in the lab where we can do serial dilutions and we can do streak isolation or we can do pore plates. That can be very time intensive because you have to get in the right dilution range, and if you have to make the media, that's time consuming, and then you have to plate them out, count the number of colonies, then you have to dispose of the plates. It's very, very time consuming. So the whole idea behind using a spectrophotometer, it's an indirect way of counting the microbial growth. But you have to correlate the numbers that you're going to get from this machine with, okay, great, I got this number. What does it mean? Correlate it with the number of bacteria. And how do you do that? With whatever media you're using, you're going to have to run one run through to come up with this correlation or this formula. In other words, you're going to have to take a sample. You're going to have to monitor it, get a reading, but at the same time, from that same sample, you're going to have to plate it out and be able to say, okay, excuse me, and you're probably going to have to dilute it out, so you're going to have several different dilutions, count your colonies between 30 and 300, so you got to get in that range, and be able to say, okay, this sample has X number of bacteria per mil. Once you know that correlation with that particular media, and if you change media, you have to redo everything because it's specific for just that one media. But once you've set up this correlation, okay, I know if I get a reading of 0.375, I can plug that in and calculate how many bacteria I have. Once that's done, then it's very easy. You just put the sample in, you close the lid, bingo, there's your number. Next sample in, close the lid. This lid closes this down, so you always close it before you take the reading. And it's literally that fast. Just put your cuvette, which is what the sample holder is called. Put it in here, close the lid. Bingo, there's your number. Next sample. Bingo, there's your number. Next sample. Bingo, there's your number. You don't have all of that time making the media, incubating it, counting the colonies, now having to sterilize all the waste, etc. So the advantage is ultimately it's going to be much faster. So how does all this relate to your worksheet? Here we go. On your worksheet, it starts out about calibrating. How do you get this relationship between the number of bacteria versus the number you see on the spectrophotometer? So this is an example just using the bacteria Pseudomonas aeruginosa. And somebody set up, this is actually taken from someone's research. And so the very first part of it is you have your stock solution. You probably know that that's pretty concentrated, so you need to dilute it out. So on the first page, you are just doing your regular serial dilutions. You've already done that. Then from the last 3 tubes, for each tube, you took basically 3 samples, and you plated them out on Petri dishes. Now remember, watch, you're only taking 100 microliters. 100 microliters is the same. Let's see if I do this. So 100 microliters is equal to 0.1 milliliters. So if you have a tube, and let's say this tube, you already calculated, is a 1 to 100 dilution. Remember, if you take 0.1, there's supposed to be a point in there. sorry, my thing's messing up a bit. 0.1 milliliters, which is the same as 100 microliters. And you put that on a plate. Remember, that is like doing another 1 to 10 dilution. So this plate, instead of being a 1 to 100, it's going to be 1 to 1,000. And so that's what kind of part 2 on page 1 is just asking you to do. You have set A, set B, and set C of plates. You're taking those from the last 3 tubes on the far right. And it's wanting you to figure out what is the dilution on each of those plates. Once again, you guys have already done this with the dilutions. With now the second page, it's showing you incubated those plates from page 1, page 2, set A, B, and C. Those are the same plates. It's after incubation, and you see all those little dots on there. Do you have to count all those dots? Yes, I'm sorry. You do. And so you count them, and then you get the average for each set. This is letting you know, or how you figure out, which one of those sets of plates are you going to use for the calibration for the rest of the experiment. You're only going to use one set because remember, we only count between 30 and 300. So you're going to have to count those and figure out what you're going, which set it's coming from. And then at the very beginning on page 3, it says using the plate counts from the usable set, that's from page 2, calculate the CFU per mil in the stock. So CFU stands for colony forming units. That is what I know Dr. McClinton tends to use. A lot of people use that. And then a lot of us also use a different term, but it's the same thing. I tend to say how many bacteria per mil. It's the same thing, okay? So the first question on page 3 is just asking, using the plate counts from that usable set, figure out how many bacteria you had. So remember, just as an example, this is not the answer to the problem on your page, but just as an example. Let's say on this plate right here, let's say we had counted the number of colonies and you ended up with, let's just say 147 colonies on this plate. And I'm sorry, I am not used to writing on these tablets, so I know it looks really messy. I apologize. So if you counted 147, you can count that because it's between 30 and 300. And all you do is you take that 147 that you counted. And remember, that's what we're counting on here, as though there are all these colonies. I'm not drawing 147 on them. But just pretend there's 147 on there. So you take that 147, you multiply it by this 1,000. That's the dilution factor. So that times 1,000. And you end up with 147,000. This is bacteria per mil, or CFU per mil. That's how you calculate. That's all it's asking you to do at the top of page 3. Is from the set that you counted on those plates that was inoculated with the dilutions from page 1. So you have to go in order on these pages. You can't skip around, okay? Because you need page 1 to do page 2. You need page 2 to do page 3. Okay, so that should get you through the top of page 3. Now the next question is, just partway through, it just says, now determine what the CFU per mil, remember that's bacteria per mil, would be in the absorbance of 0.100. I'm going to flip to a new slide. So here we go. You came up with a number. Let's just say that number you came up with at the top of page 3, where I did the sample and just said we had 147,000, okay, bacteria per mil. We'll use that as the example. Now, back on page 1, at the very beginning, it says the absorbance, or the OD at 600. That's just telling you OD 600. That's telling you what the wavelength was that 600. Of that initial suspension was measured at 0.892. So it's saying that when you measured this, you got a reading of 0.892. Where did this number come from? This came from back when you put that sample in the spectrophotometer, and I told you how you close the lid and it gives you the number on that little screen that I showed you. This was the number that came up there. This is showing you, this would be the absorbance, which means you're measuring the amount of light, ultimately the amount of light that did not pass through for absorbance. Transmission is where you measure the amount that went through. Absorbance is the reverse of that. So now it is saying in this question, what would be, let's see, what would be the concentration, and this is going here. What would be this if the absorbance was 0.100? How many bacteria would be? If that absorbance was so much less, how many bacteria would it be? So to calculate this, and I know it confuses people because the person who wrote this came up just trying to come up with another example analogy of how to do this. I'm just going to show you. What you do is you take 147,000, did not mean to do that, bacteria, yes, bacteria per mil, but you're going to take this, and you can, do you remember how you can cross multiply? And this will give you x, which in our case is what the question mark is. Okay, so we're solving for x. So now, if I go ahead and I have my calculator handy, so I take 147,000 times 0.1, and then I divide that by 0.892. That is going to give me a number of 16479, depending on how far out you want to go, say 0.8. That is what x is equal to. Okay, that's all it's at. So this would be bacteria per mil. Which, look at your answer and doesn't make sense. That at, let me switch to a different color here. At this higher level of the 0.892, you have this higher number, 147,000. But at a reading of 0.1, which is a lower number, then the number of bacteria per mil should be lower. So you can kind of double check your answer there a little bit. So this is when I talk about setting up an equation where you can just figure out that relationship of how many bacteria, relationship with the weight, with the absorbance reading at a particular wavelength. This is what I am talking about, is this relationship right here. So that no matter what reading you get, if you put another sample in and you get 0.292, you would put that here. So does that make sense? Let's move to a different slide. Let's just say you asked what was, you have a third sample and you say the reading is 0.3, it's supposed to be a 357. Then now you just take 147,000 bacteria per mil. I always say keep your units so you don't forget what you're doing. A friend of mine, a colleague of mine from years ago that used to teach chemistry, he used to always tell his students, you cannot have naked numbers. Always keep your units. Okay, so now you're going to be going, okay, what is this if I have 0.357? So once again, take 147,000, that's a 0, your bacteria per mil times 0.357 and divide that by 0.892. Now, think about this for a second. The original number was 0.892 and you had 147,000 bacteria. When we did 0.1, we had about 16, a little over, between 16 and 17,000. So if 0.3 is between 0.1 and 0.892, you would expect the number to be between those two. This is the way you just kind of mentally double check yourself. And once again, I'm using my handy dandy little calculator, and the answer is 588,333. Once again, bacteria per mil. So yes, the answer did indeed come out between the two. So that's how you would calculate the answer. It's about in the top third on page 3. It's before, and people I know get confused with the apples in the basket. She was just trying to do an analogy there as to how to set up proportions. Okay, so then we move on to page 4. Page 4 now is going